Get started, and I'm going to note some of my institutional affiliations before I really dig in. So I'm in the Department of Geosciences here at Stony Brook University, and sort of formally, as part of my hiring here in 2016, was hired with the understanding that I would, whoa, okay, this thing's a little weird. That I would work very closely with the Turkana Basin Institute, and I'll come back to that a few times during my talk. I also thought it would be a good chance to mention that colleagues in the School of Marine and Atmospheric Sciences here sort of graciously in the first couple of years I arrived on campus allowed me to sort of gain affiliation. As Turi mentioned, stable isotopes are everywhere, and including in the ocean. And so lots of applications and wonderful student collaborative projects have come out of SOMAS. And Paul Shepson, the dean of SOMAS, was here, and I see a few SOMAS faculty in the audience and students. And I'll come back to some of the work that we're really starting in collaboration with SOMAS scientists to study the actual lake, right? Everyone's been focused on things that aren't the lake, but almost everyone has shown a picture that includes the lake surface, right? So big lake in the middle of the basin, we need to study it. But before I dig in, I want to make a little bit of a disclaimer, which is on, what's today, the fourth day, midway through the fourth day of the conference, it strikes me that it becomes really hard to say something original. Especially when your talk follows one of the handful of stable isotope geochemists in the National Academy. So thank you, Turi, for giving an introduction to the talk. But I will say that my one advantage is, with maybe the exception of President McInnes, maybe Pat, and now you all who've given a talk up here, I've actually been up here once before. It was during COVID. So there was no live audience. I actually gave a talk to a bunch of Zoom screens in the audience, but nevertheless, I was up here. So I'm used to the bright lights. And as part of that, I received some training in giving a public talk, right? Not gonna claim that I'm an expert in it, but nevertheless, I was trained by a wonderful group of colleagues and people here at Stony Brook University through the Alan Alda Center for Communicating Science. So I have some tricks. So they told me that I need to move around, so I'm gonna try and do that. Most people have come up here and just stood over there, right? So actually I gave my talk mostly last time from over here. They also told me to use devices that will engage you all, keep you all engaged, okay? So thankfully everyone just filled up on coffee, but if you are tired in day 4, you would be excused, right? So what I'm gonna do, or what I've decided to do, is say dope, like Homer Simpson, every time, or just before I say something that has already been said before in the last few days. So, dope. I think it was Johannes on day 1, I don't know where Johannes is, who pointed to the sort of immortal words of Richard that live on the Turkana Basin Institute website. And my referencing them, or a sort of snippets from the full text, comes with a story. And the story goes that the search under which I was hired was started relatively late in the cycle of sort of academic faculty hires. So I was a sort of ambitious postdoc and had a bunch of applications in, and I received an email from a friend and now very close colleague, Troy Raspberry, saying, hey, Greg, you need to apply for this job. And I thought, cool, and like many of you, I mostly knew of Richard through stories that I had heard from my PhD advisor, Ben Passy, and folks like Naomi Levin and occasionally Turi. But I had never met Richard. And so I only really knew Richard from the pages of National Geographic. So the first thing I did, after I can literally picture me sitting in my office as a postdoc, receiving the email from Troy, clicking on the Turkana Basin Institute website and navigating to the page that says, I think it says, about TBI or a message from Richard Leakey. Oh, I should click that first, okay? And I pulled it up, and this was, I mean, I didn't really need much convincing to apply to the job, but I read these words: a science that is on the move. Each year, more discoveries are made as a consequence of our understanding that changes. And that TBI, the promise was that there was an opportunity to facilitate new explorers' discovery of treasures buried in the Turkana Basin. And that was Richard Leakey's dream, right? So I found it just the cherry on top in terms of motivation to apply and put every effort into the interview process. So thank you, Troy. Thank you, Richard. And thank you to the whole sort of TBI family who's welcomed me in. So in my mind, Richard's legacy was embodied by another sort of big figure in the TBI ecosystem, Isaiah Nengo, who, like Richard and Lawrence and others at TBI, sort of welcomed me. The first time I met Isaiah, he was sort of this like unsure figure hanging out outside of my lab door, which many of you saw the lab on Monday morning. And he sort of came in and he busted in the lab and talked all about the Miocene and how he wanted to reinvigorate research. And so, you know, it was obviously a really tragic loss and sort of a double whammy to have Richard pass and Isaiah leave us soon thereafter. And so a lot of the work that Kevin described yesterday, and you'll hear more about this afternoon and tomorrow, is the sort of brainchild of the Turkana Miocene project in the sort of memory of some of our work recently is done in the name of Isaiah. Okay, so I guess dope, right? So Kevin showed this slide yesterday, which really just articulates the sort of ambition of the Turkana Miocene project. Some of the leadership, or I guess all of the leadership team, and articulates or makes the point that, you know, there are 50 members from, you know, 15 to 20 institutions. Some of that is sort of changing actively. We're adding and losing new institutions. It's a really fun, highly collaborative effort. I don't know if Kevin noted yesterday that we're sort of present online. So you can find us on Twitter and Instagram, and we'll try and update things as we embark on this summer's field season. Here's that big group. As Kevin said, you can sort of find your friends, find your colleagues in this list. But I wanted to show it, and it maybe is a little hard to tell, but there are names that are sort of being highlighted here in bold and italics who are going to be featured in a lot of the things that I'll talk about today. Okay, so just to give you a sense of temporal scale, and some have talked about various parts of the Miocene, but sort of the circled sites, this is borrowed from Craig Feibel, outline these sites that are outlined sort of show the sequence of our project and the sort of various ages of sites in the Miocene in the Turkana Basin that we've been visiting. And with this Venn diagram, I was trying to articulate this sort of like tools, right? So I'm talking about stable isotope geochemists, as Turi said. We like tools, right? We like, you know, forget the analogy that Turi used, but you know, we like cars, Ferraris, right? That are supposed to make the right turn on Turi's sort of left-hand, right-hand turned road picture before. And so these are some of the tools that our project is bringing to bear on the Miocene climate, Earth, and life record. And the sort of idea with this sort of highly overlapping Venn diagram is that many of these tools are sort of answering questions that really span the sort of disciplinary boundaries of the project. And of course, you know, leave it to the isotope geochemist to pull down something and show it to a bunch of anthropologists from Encyclopedia Britannica. maybe audacious of me, but this is the motivation. I don't need, I've given this a form of this talk before to my geologist friends, and they don't know that I'm sort of pulling one over on them, but really this is the motivation, and I'm preaching to the choir here. And then there are things like Isaiah's discovery of Alessi at Napodet, a sort of mid-Miocene site. And if you read that paper carefully, many of you were on that paper, you sort of pick up words and sort of ideas that really call on the need to bolster and reconstruct paleoenvironment during this time period. So we can also look to the existing data, as we've seen many examples in the last several days of climate both at the global scale. So what I'm showing on the left here is PCO2, the sort of paleo-CO2 reconstructions, side by side with a very local, as Kay was sort of alluding to in her talk, this sort of local expression of some global climate phenomenon that is unfolding over several million years. And the thing I like to point out with this is from a paper by Naomi Levin, a review paper that I think puts a nice stamp in time in terms of what the record looked like from 2014 to 2015. And what I point out when I show this slide is that the record here, as shown, stops right before all of the interesting stuff I showed in the previous slides, and some of which was talked about earlier in the conference. Okay, and Turi did, you heard it from the guy, right? So I don't need to explain the virtues of oxygen isotopes, but Turi did a very nice job of explaining some of the problems, okay? So we have this sort of big global record of paleoclimate that comes from deep sea cores and little shells that you can extract from those cores. And they tell you something about how, the ocean is a big, huge heat sink with a lot of thermal inertia. And so it takes a lot to change the temperature of the bottom of the ocean and to sort of superimpose squiggles on this line here. And so we can do this sort of scale hopping over to the record for where all of the action is happening over the last 10 million years in East Africa, our area of interest. And so this is shown in soil carbonate oxygen isotope records that have been generated prior to 2015. And again, that scale stops at 10 million years. So when I first started working on this project, I was like, okay, well, it seems like the need is pretty clear, and all we need to do is sort of extend these records as if it were that simple. But Turi also really nicely laid out, and now I don't have to talk about it, that the oxygen isotope record can be interpreted two ways. So it can be interpreted as a paleotemperature record if you were to assume the oxygen isotope ratio of the fluid from which these carbonates were forming, or it can be a record of the oxygen isotopes of the fluid if you assume a constant temperature. Okay, so Turi said it, right? All we need is a paleothermometer that is derived from these same materials, but is independent of a temperature signal independent of the oxygen isotopes. I was going to make a joke about you all now becoming students in my stable isotope geochemistry class. So what you're seeing here, I'll spare you a joke. What you'll see here is a carbonate lattice that I've sort of populated with all of the elements that are involved in a calcium carbonate, right? These calcium carbonates are making up those soil minerals. So the greens here in this lattice are calcium groups. The oxygens found in groups of 3 are the oxygens in the carbonate groups, and then the carbons are at the center of the carbonate group. Okay, and if you're a chemist, you look at this and say, oh, cool, maybe you're interested in the structure or the mineralogy of it. But us isotope geochemists see the world slightly differently. So now what I've done is taken those carbon and oxygen groups and sort of color-coded them where they are substituted by a heavy, rare isotope, either oxygen 18 or carbon 13. So you can see that in some molecules, we have a carbon 13 bound to just a regular old oxygen, most of which is oxygen 16, or an oxygen 18 bound to a regular old carbon 12, okay? Most of the science in the last 60, 70 years has focused on just understanding, just sort of counting up all of these isotopes in a material and making the carbon 13 to 12 carbon ratio. But the advent that I'll be talking about today and what we've been spending most of the last 5 years on is the cases where you find a carbon 13 and an oxygen 18 bound together. And as Turi said, this is a signal that is entirely dependent on thermodynamics. It doesn't care about how many 13s or 12s or 16s or 18s are in the mineral. So to give you a sense of the challenge that this presents, right? So you've got two rare things together in the same group, okay? So to give you a sense for how challenging that is, Turi mentioned patience, okay? To measure carbon 12 to carbon, or carbon 13 to carbon 12, that's like measuring the amount of nitrogen in this room, in the air, okay? Most of the air that we're all breathing is nitrogen. Thankfully, some of it is oxygen, or else you'd all really be asleep. To measure oxygen 18 to oxygen 16, that would be like trying to measure the approximately 1% of the air that we're all breathing that is a noble gas, argon, okay? That's about 1%. To measure these clumped isotopes, the CO2 in this room is probably, with all of us in here, is probably something like 1,000 parts per million, okay? 1/10 of that would be like measuring one of these carbon clumped isotopes, carbonate clumped isotopes. So they're really challenging, and part of Richard's legacy through TBI was sort of giving the Department of Geosciences this sort of excuse to buy into some of this very exciting science. And Craig Feibel yesterday showed a picture of Ian McDougall next to his mass spectrometer, and I was like, yes, okay, now I can show this sort of like modern version of that. So this is the mass spectrometer that we've sort of built, constructed in our lab, and a prep line that is specifically designed. You can't go, to like Fisher or VWR and just say, give me one of these things that can tell me what the clumped isotopes are. We have to make that ourselves and spend lots of time purifying CO2 so that we can pick out that very, very rare combination. Okay, so I'm going to prove to you very quickly that it works. My time is vanishing. So the details of this line are unimportant, but what you should see or take away is that there are data that fall on the line, and there's not a lot of scatter around the line. And the relationship here is between temperature on the y-axis and the clumped isotope ratio that we measure, temperature on the x-axis and the clumped isotope ratio on the y-axis. And this is effectively a transfer function. So we can take this to various modern or contemporary soils, and there are a number of us in this sort of very small subfield who've been working on this, and ask the question, if we generate a clumped isotope temperature or oxygen isotope estimates of the water, do we get the right answer? And so the fact that lots of this data falls along a one-to-one line gives us some indication that we're sort of safe in applying this to paleo sort of applications. Turi already mentioned some of the work that he and his student Ben Passy and Naomi Levin did in 2010 which really put a mark in both the clumped isotope community, with Turkana being the first application of this sort of highly specialized method. And they sort of made the case that Turkana, perhaps not surprisingly, has remained hot for at least the last 4 million years. I will also make an observation that not only is Turkana one of the hottest places, but in a series of papers that came out in the last year or so by Callum Munday and colleagues have really pointed to the fact that Turkana is unique in its aridity or dryness, right, as well. So this little red dot, Chalby can stand in for sort of northwestern Kenya here. But this is the latitude of the deserts on Earth, of the major deserts on Earth. And you'll see the sort of lowest latitude desert in the tropics there's really only one, and that's the sort of part where we all are drawn to. Okay, so we're extending the record back into the Miocene. I'm almost nearly out of time, so I'm going to skip through this relatively quickly. The first place we worked at was Lothagum. We are selecting pedogenic carbonates and developing a sort of chronology of where those are from in the sequence. Most of these samples were collected during the 90s effort by Craig Feibel. And here's the record, right? So the blue data are the data from Ben and Turi's work published in 2010. And the red data are our effort to both sort of prove that we can replicate their results in the Pliocene, but then effectively extend this record back into the early Miocene or the late Miocene. And the bottom line is that it's sort of the temperature story, at least, isn't all that interesting. Temperature appears that the Turkana Basin appears to have been hot for at least the last 8 to 9 million years, or as hot as it is now. And therefore, any changes that we see in the carbonate oxygen isotope record must be driven by changes in soil water oxygen isotope ratios as opposed to temperature. And so here is this sort of backstripped, this bottom figure here is the backstripped fluid delta 18O values. We'll come back to that. I'm going to skip through this for sake of time and jump over to a sort of compilation where we have temperature on the top panel here, fluid oxygen isotope ratios in the middle, and then the soil carbonate carbon isotopes, which come along for the ride when we make these measurements on the bottom panel. I'll sort of assert, and we can talk about this later today, that I think this sort of record makes a lot of sense with the work that was published by John Wynne in the Lothagum monograph on these sort of distribution and types of soils at Lothagum. And if you sort of squint your eyes and follow along with me, we can sort of start to superimpose some of the global climatic events. So there was a change in the carbon isotope ratio of these benthic foraminifera in the late Miocene associated with an increase in marine productivity during that time. The onset of that is sort of coincident with the earliest part of the record at Lothagum, and the local maximum, at least in carbon and oxygen isotope ratios, coincides with a change point in that, in a statistical analysis of that benthic foraminifera record. The values decrease thereafter in both carbon and oxygen isotopes and reach a sort of local minima during the height of mid-Pliocene warming, and sort of increase thereafter, implying a sort of more or increasingly arid and increasingly grassland-dominated landscape. Okay, but here's a little dirty secret. If you look at these samples in cross-section, so this is a cross-section through one of these carbonates, they're not just sort of beautiful, like you can see all these, they've got inclusions in them of detrital minerals, and then they're sort of crossed through with all of this secondary carbonate that presumably grew in sometime after the original nodule grew in the soil. So I'll draw a little line, and it may be hard to see a little circle on there. That's about the scale of our sampling. And so one of the things that we're working on currently is a method to essentially take out the secondary overprint from these pedogenic carbonates. So we're working on statistical methods for segmenting these images. I said I'd walk around. To segment these images of these petrographic images, and this is largely the work of May Saslaw, my graduate student, who's unfortunately not here today, but hopefully you got a chance to meet her. And May is working on a sort of new method, a new approach to try and make this sort of fidelity of these carbonate records even better. So instead, she makes sort of Swiss cheese out of these. Instead of just subsampling once, she makes kind of Swiss cheese out of these carbonate nodules, and then we can map on where she sampled, we can map onto the petrographic image to come up with individual measurements of the relative proportion of sort of primary versus secondary carbonate in these samples. And Turi talked a lot about sort of mathematical methods. I paid attention in my linear algebra class, and a simple linear inversion of the data. This is just an example from a pedogenic carbonate from Baluk. Shows that we can sort of successfully remove what we interpret as a primary signal, so a paleo temperature of 37 degrees and a carbon isotope ratio of about -9 per mil from this sort of diagenetic overprint, which shows a much warmer temperature and slightly heavier delta 13C and delta 18O values. I'm going to skip through, but I'm happy to talk about the sort of climate modeling comparisons that we're going to try and make as a part of this project. But I wanted to end by showcasing a couple of things that are kind of on the horizon. And Troy Raspberry this afternoon is going to talk about the sort of application of pairing these carbonate clumped isotope measurement with dating of things that aren't pedogenic carbonates, but that are nevertheless useful for understanding this sort of stratigraphic chronology in the Turkana Basin. One of the really exciting projects that is just beginning with seed funding from Stony Brook University and the School of Marine and Atmospheric Science is led by a graduate student, Madison Mule, Josie Aller, who I believe is here, and our colleague Mike Frisk, trying to extract isotope information from fish teeth so that we can develop a record of paleo temperature and oxygen isotope ratios from Lake Turkana itself. So here's some of our preliminary results. They're actually quite exciting because May, my graduate student, has also been working on a massive compilation of water-oxygen isotope ratios from the Turkana Basin, which show a big difference between, so temperature was about the same 1.6 million years ago in Lake Turkana, but the oxygen isotope ratio of that lake water was 5 per mil lower than it is, 5 per mil lower than it is today. So what determines the lake water oxygen isotope ratio? And it will be of no surprise to you all that it's evaporation. So evaporative processes fractionate oxygen and hydrogen isotopes. And so what you're seeing here is a compilation from May of lake water oxygen isotope ratios showing the enrichment that follows from this sort of closed basin evaporation. And then finally, as Turi alluded to, there's sort of the third oxygen isotope, which is even more elusive than the oxygen 18, oxygen 17, predictably fractionates also with evaporation. And so one of the exciting things, this is work I wasn't involved in, but led by a colleague, Julia Kelson, showing the triple oxygen isotope ratio of pedogenic carbonates from all over the world, mapped onto their aridity index. And I've been able to generate some in Ben Passy and Naomi Levin's laboratory at the University of Michigan. We've generated some preliminary data that shows modern East African soils plot along where you might expect them to. These are data from Impala collected last summer. And interesting, some of the soils from the Miocene sediments at Lothegum plot much lower, and which is implying this sort of more arid landscape. With that, I'll wrap up. Flash this picture, a dope. Kevin showed this photo previously. I'd like to thank NSF and the Turkana Basin Institute for providing funding for a lot of this work. And I'll make a plug. So one of the exciting things I think that should come from this conference is more opportunities for all of us to gather. And so May Saslaw is leading, along with Emily Beverly and Bill Lukens, those names should be familiar to many of you, a session at GSA this coming fall. I was told by May to note that Kay has been invited as the speaker, but I don't believe Kay has accepted, and there is no pressure. There's no pressure. It was very explicit. There's no pressure. But if you feel like you have something to contribute, I think we would all welcome to hear it at GSA in the fall. Thank you.